Hey everybody, and welcome back. We're here to talk about, we're still Unit 5, we're still Lesson 1, but now we're going to be looking at Exploration 2. So we're looking at the second part of Unit 5, Lesson 1. So really what we're going to talk about now is, in Exploration 2, is how are cells going to divide, when do they know when to divide, and kind of what size of cell is going to be the perfect size. So a typical animal cell grows only to 10 to 20 micrometers. And the reason it says only that is because the cells aren't going to get any larger. If they continue to get larger, then they're going to divide. And the reason we're looking at is the surface area to volume ratio. And what I want you to know is that the larger the surface area to volume ratio, the more objects things are going to be able to bring in versus their volume. So we want a high surface area to volume ratio. And what we're going to see here, and I'm not going to get into really the calculations of it, but what you're going to find out is a smaller cell is actually going to have a higher surface area to volume ratio. And that's why cells are going to remain small. So as we scroll down here, we're going to talk about cell size. So no matter how materials move across the membrane, they must be transported in adequate amounts and with an adequate speed to maintain homeostasis. So it's very important that these molecules that need to move across a cell membrane, things like water and sugar and carbon dioxide and oxygen, these things need to be able to move quickly. And if the surface area to volume ratio is low, it's going to need more of that thing than we can get in. So it's going to need more oxygen than we can get in. So it's important in that surface area to volume ratio that we can get things in quickly. Okay. It says if there's not enough surface area for materials to cross in and out of the cell, then the cell might not be able to absorb enough materials or get rid of waste effectively. And that's why surface area to volume ratio is important right there, that little paragraph. Okay. Now let's talk about how it's going to regulate cell division. Regulate means to stop or control. So cell division, what we're talking about is how are we going to control cell division? When does a cell divide? So really what we talk about here are the external factors that are going to regulate a cell and the cell cycle. It can either be a physical signal or a chemical signal, right? It says it right here, physical or chemical signal. One example of a physical signal is going to be cell-to-cell contact, and that means that cells have an ability to sense one another. So what's gonna happen is individual cells will divide until they touch one another. And then once they touch one another, they know that they are out of room, and then they're going to stop dividing once they touch each other. Another way that they're going to know is by chemical signals, and they're going to release hormones or growth factors or different proteins that are then going to stop cell division. So there's several ways that your body knows I need to create more cells versus I need to stop. And we call that cell regulation, cell division regulation. Okay, so as we look at the next one here and we scroll down, the next term we want to talk about is apoptosis. And what apoptosis is, and I love this definition, it's one of my favorite ones, it's programmed cell death. I feel like that should have like some sort of like Darth Vader entrance sound. But so apoptosis is going to be controlled cell death. And what that means is the cell is just going to kill itself, or the body is going to kill that cell. And it could happen for many reasons. One, you can have damaged DNA. And if that cell has damaged DNA and we allow it to survive, then it's going to copy and copy and copy. And now that one cell is going to turn into hundreds, thousands, millions. Who knows how many cells are going to be created. And a lot of times we call that cancer. That's how we get cancer. Or it's just not going to be needed for the body anymore. You know, our many organisms, whether they're human or other living organisms, are going to go through growth, and they're going to go through development. And when organisms develop, sometimes they no longer need those old cells, or we're just going to make new cells to replace them. And those cells are going to go through apoptosis, which remember is programmed cell death. It's going to die. Okay, and now we get to cancer, and cancer is one of the leading diseases that we have and that we're facing right now in science. And this is the common name for a class of diseases characterized by uncontrolled cell division, and that's the important part here. The uncontrolled cell division is important because these cells we do not want to divide. We don't want them to go. And so what a tumor is, and we'll kind of click on this, a tumor is cancer cells that divide more often than the healthy cells. So what that means is that we're going to have these cells right here, and these are healthy cells, and these are healthy cells. What's going to happen is a tumor, these cancerous cells, for whatever reason, are uncontrollably dividing, and they're doing it at a much faster rate than these normal cells next to it. And it's going to cause this bump, or it's going to cause the growth. What will also happen is these cancer cells will start to get in the way of the regular cells, and now these regular normal cells are going to start dying. Another thing that's important to know about cancer is that these cells don't necessarily perform a function, right? And so what happens is, it says right here, cancer cells are hazardous because they do not perform the functions of the normal cells. So for example, if this is a skin cell, they have jobs. And this tumor right here is not going to do the same job as these healthy cells, right? If you have a cancerous cells in an organ, for example, your liver, your kidney, your heart, really anywhere, we can find cancerous cells and tumors anywhere. Those cells aren't going to perform the function that we need these cells to do. And therefore, that's why we see that cancer causes death in a lot of individuals, is because where they find the cancer, we need those organs to work, whether it's your liver or your kidney or your pancreas or your heart, or your lungs, right? These are all places that those cells do life-saving functions for us. And so if they stop, then we're gonna be in trouble. Okay, so that's kind of how cancer is. Now that was exploration 2, and now I want to move on to exploration 3. We're going to do 2 explorations in the same lesson here, and this is really a quick exploration on the history. Okay, so the first that we talked about is Robert Hooke, and Robert Hooke is going to create a 3-lens microscope, and he is going to look at a piece of cork. This is Robert Hooke's microscope and the cork that he looked at, and he was the one that gave the word cells. And because he thought that these little these cells looked like the cells of a monastery where he lived, a monk's monastery. Also, it's important to know that it took nearly 200 years to make the link from cells to then all living things are made of cells, or cells are required for life. So here's a microscope that he would have used back in the 1600s. And then this is a microscope that we use today, a compound light microscope, and what we can see and the magnification we can get to. So as we scroll down, the cell theory is going to be the main thing that we talk about. And here are the people that help get us to the cell theory. So Zacharias Janssen was an eyeglass maker who used lenses to create the first compound microscope with 2 lenses. Then we're going to get into Robert Hooke, and he made a 3-lensed compound microscope. And he also was the one that we just talked about that named them cells. Anton van Leeuwenhoek is a person he was a Dutch tradesman, but he developed an even more powerful microscope, and he was able to look into water, to pond water, and see microorganisms, single-celled organisms. So he was really the first one to see, or at least to document seeing living organisms in water. Matthias Schleiden, he was a German botanist who basically said that plants are going to be made of cells. Right, and so he was able to take these microorganisms in water and then also attribute them to plant cells. Theodor Schwann is the gentleman that's going to find the links between plant cells and animal cells, and he's going to say that they are very similar. And he concluded that all living things are made of cells. And this is very important to Schwann, is that he said that all living things are made of cells in the cell cycle. And then Virchow is going to be the one that's going to talk about all cells come from other cells. So Virchow is going to have that third point where all living cells are going to come from previously existing cells. So as we take the information from these 6 men, we're going to come down here and we're going to get to the cell theory. And the cell theory, by definition, it states that organisms are made of cells, cells are made from other living cells, and they're the basic unit of life. And here is those 3 parts of the cell theory. It's important for you to know these. These are something that you're going to need to be familiar with on the test. So all organisms are made of cells. All living organisms are made of cells. All existing cells are produced from other living cells, meaning they come from pre-existing cells through a process through the cell cycle and mitosis and making new ones. And then the last part is the cell is the most basic unit of life. If you go smaller than a cell, you no longer have a living organism, you have some organelles, which are parts of that, but cannot sustain life on their own. So that's going to be the end of Unit 5, Lesson 1. So can't wait to see you guys again, and we'll start Unit 5, Lesson 2.